Welcome everyone to BioC 2020. It is a great pleasure to bring to you Bioconductor's first ever virtual conference, and I'm just going to give some brief introductions to the conference format, and following that, Martin Morgan will give an overview of the Bioconductor project and recent happenings more in general. So I just wanted to start with a few notes about our Code of Conduct. You can find this under the Code of Conduct tab, which I recommend you taking a look at, and you'll find here also where you can report by email or anonymously any violations of the Code of Conduct. But I'm going to give you just the broad overview here. First of all, no harassment. This part is similar to what you would find at most conferences. And just as a general rule, if I have to summarize in one line, I recommend keeping your focus on software and science. That's really what most people are here for, as opposed to starting to talk about or point out individual characteristics, certainly not getting into race, gender, anything like this. So let's keep it about software and science. The second two are kind of specific to this conference, so I just want to point them out to everyone. This is a public forum, and our social media policy is that content presented here may be posted publicly on social media. So if you're a presenter, please keep that in mind and don't present secret things that you don't want to be public yet. If you don't want it published on social media, make sure to let people know that, but I do recommend that you only present things that you want to be public. Furthermore, the sessions will be recorded. They will remain on this platform for a year and eventually posted on the Bioconductor YouTube channel. And then finally, we want to maintain a welcoming environment, and this means no bullying of any kind, intellectual bullying, belittling, making fun of others. And that's just because this is the kind of environment we want. We want it to be welcoming to everyone, including beginners. And I just want to emphasize this applies to all online forums, including GitHub, Slack, the support site, mailing lists, etc. A little bit about the Pathable Platform. I just want to highlight a couple of things. You'll probably discover most things on your own. But make sure you take a look at the Conversations public forums. That's where we're going to post key communications, such as links that you won't find elsewhere, such as to the Slack teams, poster sessions, lightning talks, and other things. Another little tip that I'm just mentioning because I found very useful is that under the search button, there's something called track that will allow you to narrow down the program to workshops, birds of feather, talks, etc. Makes it easier to find things as you build up my agenda. Don't worry about sessions filling up. You do have to add them to your agenda in order to attend, but there are as many seats. There are 500 seats and 500 attendees, so the sessions will not fill up. You can ask and upvote questions in the polls tab, and you can do this during a session or in advance of the session, and moderators will use this to take questions for the speakers. And then after a session is over, recordings should become available about 15 minutes afterwards and remain available for a year. Now, other than in the conversations, there are some ways to talk face-to-face. You can use the Pathable's schedule meeting and want to meet features. We also have these poster rooms that you'll see a link to. I'll talk a little bit more about them, and they will remain open throughout the week. If you need to contact the organizers of the conference, here is our email address. And if you need technical support about the platform, please email this virtual@conferencedirect.com. And we do have technical support for the entire week, and they are very helpful. And there will also be live support hours at 9 a.m. and 1 p.m. each day. A big part of all Bioconductor conferences is the workshops, and these are hands-on sessions where you can try out code and learn about them from the authors of the code. And we have a new format this year that we've never done before, but it's pretty cool. It's one where not to get into too much geeky stuff here, but every author created their own GitHub repo as an R package, used GitHub Actions for testing and building, packaged down to create a website with all the built content for the workshop, and a Docker container that allows you to run it on the cloud. And during the conference this week, you will be able to run all of these on the cloud at workshop.bioc.cancerdatasci.org for free, thanks to the Cancer Genome Consortium. You can also run them on Docker locally, and there are instructions for doing this on our public BioC 2020 page. And just as a note, you can also run any of these on a server, such as a high-performance computing center using Singularity. And if you go to this public page, you'll see a list of the workshops with links to their web pages and to their GitHub pages and Docker images. And I want to thank for this, Sean Davis was really the magician who imagined this whole new system and created it. It's really something interesting and new, so we look forward to hearing your feedback on it. And Mikhail Dozmarov, who was sort of the muscle behind a lot of this. If all of the workshop authors by now are well familiar with Mikhail for having communicated all of the issues with their workshop and sent out all of the related communications. We also have a novel format this year for posters. Jian Lu has created a gateway or a gateway area on F1000 research for us. So all of these posters are available there. And then Nathan Sheffield created a novel format for the poster session, which is really great. Once you go there, you can fill your own shopping cart with posters that you're interested in. You can easily browse them there. And there are also general discussion tables, and then there are discussion rooms for each poster, where during the poster sessions, you'll be able to go and talk to the poster authors. We have a big program that is keeping us busy 8 hours a day all week long with very few parallel sessions. 23 workshops, 24 contributed talks, and 25 posters. Matt McCall coordinated all of this. Many other people contributed to the reviewing and making this happen. But Matt was, again, the person who many of these contributors have heard communications from. And then we have also a big, what I call the non-competing component, including the keynote talks, birds of a feather sessions, where people with like interests can meet and talk to each other. Sessions to meet the technical advisory board and community advisory board, which will present for the first time bioconductor awards and lightning talks. And here, Charlotte Sonneson and Simone Bell were the major forces in making all of this happen. The lightning talks, I just want to let everyone on Thursday and Friday. If you want to give one, a 4-minute talk, you should submit by Wednesday at noon Eastern time. And again, you'll find this link with all the other links under those public conversations. Just a shout out, this conference, I will tell you, developing a virtual conference and planning it is probably 2 or 3 times as much work as an in-person conference is what we've found so far. So it could only have happened without a huge amount of help from a lot of people from really all over the world. So I'd just like to thank Aideen Culhane, the co-chair of this committee, who also took care of sponsorship and many other things. Lorena Pantano, who was one of the couple main people developing the public website. Lori Shepard, also website and various other things. Raffaele Irizarry, who fulfilled many of the jobs of a local host in Boston, even though it did not end up being in Boston, and also helped us bring in sponsorship. And Samina Boca, who helped with a number of things. Those were the people who have been maybe most active, but we had many other people helping with abstract review, who will be doing session moderating and other things. And finally, I'd like to make a big thank you to Erica Feck, who honestly, it's hard to overstate how much she made this conference happen. Every other thing that might have fallen through the cracks, she took care of, and so I would say that if anything that goes right in this, I'll be thanking Erica for. With that, I'm going to pass it off to you, Martin.
Thanks, Levi. It really was a tremendous achievement getting the conference off the ground, turning it around so quickly, and organizing such a complex event. It's an amazing accomplishment. Thanks very much for doing that. So I'll just say a few words about the Bioconductor project in general, and some of this is familiar to you. The idea that Bioconductor is for the statistical analysis and comprehension of high throughput genomic data. 1900 or so are software packages developed by the core team, but actually there are more than 1,200 researchers globally who have contributed packages to the project. So the project is really the result of the efforts of people like you. It's widely used, 500,000 distinct IP downloads annually, and well respected with more than 38,000 PubMed Central full text citations. You probably know the main landing page of the project and the support site. And then many of you are becoming familiar with the community Slack channel. I just highlighted the single cell experiment channel with 208 active members. And there's a ton of activity that goes on in the Slack. And I really encourage you, especially as you become more confident using Bioconductor, to participate in the Slack. The graphic at the bottom is just one of those fun things that you can look at. These are the monthly downloads of packages from 2016 to 2020, partway through 2020. And you can see that the software is being downloaded more often each year. There are seasonal patterns, maybe corresponding to our release cycle and maybe the start of the academic year. And then for some reason, there are a ton of downloads at the beginning of this year, which was obviously the work at home culture that developed. And so people took their laptops home and had to install packages to get their work done, which is pretty amusing. I wanted to spend a few minutes on this slide introducing the core team. These are people who are paid professionals, I guess, from NIH grants to make sure that the infrastructure of the project moves forward. Kayla, Hervé, Marcel, Laurie, Nitesh, Daniel, and Jaffe, who's an RA in my group. And then there are also other people who have made significant ongoing contributions to the core of the project who are not actually my employees. Michael Lawrence, James McDonald, Andre Oles, and Mike Smith. I've tried to highlight some of the recent activities that the core team has been engaged with and that are shaping the directions that the project is going. One of them is representing large data. Hervé has been very influential in developing this delayed array framework, which allows large data to be manipulated conveniently. And Mike Smith has contributed great support for HDF5 and the HDF5 library for one type of on-disk representation. Just like in this conference where the workshops will be run, they're actually being run in the Google Cloud. I think that's increasingly the way that we'll individually do our research. We won't be installing R and Bioconductor on our laptops. We'll be using it in some other environment. And that use case will be facilitated by development of standardized containers and of cloud-based computing. And Nitesh in particular has been influential in developing cloud-based solutions and Docker containers for Bioconductor. We've invested quite a bit over the years in Annotation Hub and an Experiment Hub. This is kind of recognizing that software is one component of our computational process, but annotation data and experimental data that illustrates how to use the software is a second very important component. And the hubs separate out the large data from the package infrastructure that makes it convenient to access them. So Laurie's worked on the inner workings of Annotation Hub and Experiment Hub, and people like Marcel have contributed amazing groups of sets of resources to these hubs, including the TCGA and CBio portal. And Daniel and Kayla have also contributed quite a bit to the data that is in the annotation and experiment hubs. We work on specific packages. Kayla's worked on BioC-Set for set representation. Daniel has recently been refactoring the R track layer package to separate out basically two functions, import and export. And we're hoping that these become the standard way for accessing different file types, import a BAM file, import a CSV file, etc. It's a very flexible framework and developed originally by Michael Lawrence in R track layer, but somehow deserving of its own representation in BioC.io. Marcel has worked extensively with multi-assay experiment and derivatives, and Jaffe is kind of like our the guy on the leading edge. He's been exploring alt-rep, which is an alternative representation of the internals of R, and many other interesting emerging challenges. There's opportunities to talk with the core team on Monday, Thursday, and Friday at noon. Each of the sessions has some indication of what might be the sort of themes that will be emphasized in those discussions. I wanted to say a word or so about project leadership. We've matured over the last several years into a structure where we have a technical advisory board that you can have a chance to meet with on Tuesday at lunch that tries to establish the technical directions of the project, make sure that we're meeting the technical needs. Vince Carey is the chair, Levi the vice chair, and Charlotte the secretary. And if you follow that link, you can go to the Bioconductor Advisory Board page on the Bioconductor website and see the complete list of members and also access the minutes of our meetings. We've also been ramping up over the last year a community advisory board, which focuses on more community-facing activities like conferences, for instance, and training and outreach, and trying to bolster our representation and the feedback that people can offer to those types of activities. Matt Ritchie and Aideen Culhane lead that project. I think Laurie and Yagub are co-secretaries. And then again, others on the community advisory board are here with their meeting minutes. And I'll just mention that the community advisory board is smaller because it's newer, and we wanted to make sure we had room in subsequent years to add more members. And the idea is that every year, 3 to 5 members will leave each of the boards and 3 to 5 new members will come on board. Yes, and I wanted to mention funding sources. The project and the infrastructure that's provided doesn't come out of the blue. It's paid for primarily by grants from various parts of the US National Institutes of Health. The original funding for the project, still ongoing, is this listed as a U41 from the National Human Genome Research Institute. It supports the core team and lots of the core infrastructure. An additional grant from the NCI, the National Cancer Institute, through the Information Technology in Cancer Research program, is being very important in scalable cancer genomics research. Another NHGRI grant is funding the Anvil Computational Cloud, which in many ways is shaping what I think of as the future of computation in the bioinformatics world. And then we have some funding from the Chan Zuckerberg initiatives, seed networks for the Human Cell Atlas, and emerging open source software projects. I've placed a double asterisk beside the U41. In many ways, it's the core grant. It's coming up for renewal. It expires in February 2021. The renewal proposal has been reviewed and scored, but we don't actually know what the status of that grant is. And I've also highlighted Vince Carey's name in the Technical Advisory Board because he's taken on that leadership role in that board, and he's also a PI on the U41. And moving forward, Vince will have primary responsibility for the project's forward direction. I wanted to spend the last couple of slides. I'm falling a little bit behind, so I'll go a little bit quicker. Just thinking about the life of a package. The project's been around for quite a long time. And actually, in one of our chat rooms this morning, Kevin Roux Albrecht was talking about how he actually started working with Bioconductor and collecting functions that he made available in a package that he distributed to his lab, and then the lab said, "Hey, those are really useful. Why don't you make them more broadly available?" And that's what he actually did. So eventually, he's become an important developer in the Bioconductor and contributor to the Bioconductor community. I think this is what happens with packages, that they develop as individual research projects, or maybe more frequently these days, community collaborations. And then they get contributed to the community as a package that goes through a technical review. And then it's used in the release and develop branches of Bioconductor. And they're built nightly across multiple platforms to make sure that they're functioning as appropriate. And then one of the truly amazing things about the project is its longevity. And so packages can persist in the project for decades, literally decades. And that provides reproducibility and the ability to reanalyze older data sets. And I've got the proportion of downloads for several packages since we have the easily available records from 2009, I think. So the top line is the AFI package, which was for the analysis of microarray data. And you can see it got a 5% share of the downloads until maybe 2011 or so, where usage dropped off relatively precipitously as the community moved away from microarray analysis. Nonetheless, it's still downloading on a regular basis, still functional, still available, so that people who are analyzing microarray data at that level can still access it. The next one down is Lima, which is also used a lot in the microarray era. And as attention moved away from microarrays, it became less useful for that task. But it still has a lot of shared functionality. The methods developed for microarrays are often in many ways useful for bulk RNA-seq analysis. And so Lima has continued for a long period of time and still lots of active use. The next curve is DE-seq and DE-seq2 combined. And I think of this as like the rise of bulk RNA sequencing. And the final curve is single cell experiment, or summarized experiment, I guess, which I think of as the rise of single cell data analysis within Bioconductor. So it's really very interesting to look at this. And then, so all of these packages are still alive, providing reproducibility, reanalysis, and various stages of the life of a package. But some packages do eventually go away and are archived when the maintainer is, a maintainer is no longer available to respond to changes in R. And it's become increasingly important for us to actively archive packages and not to hold on to packages that are no longer functional beyond the willingness of the maintainer to maintain them. I wanted to talk about the life of a bioconductor, which is to say you and I on the call. And I think we all start as users, as students or postdocs or researchers relying on peers, package vignettes, help pages, the support site, and the internet in general to figure out how to get our work done. And then we eventually mature into the place where we're able to help others with our understanding and maybe with some code. Maybe we submit packages as part of an effort to gain academic credit. I see I've misjudged my time, and I was thinking I had 5 more minutes, but actually I'm at time here. So I'm going to just finish up. Maturing from a developer to a leader, and the leader contributing to organizations like this conference, but other activities. And I think it's great that we're recognizing leaders through the Bioconductor Awards, which are presented Wednesday at 8 a.m. So I'll finish there on a final note, I guess, just remembering James Taylor, who was a founding member of the Galaxy Project, a fellow traveler in the advocacy of open and reproducible science, and then really influential behind the scenes contributor to the way community funding and technical direction in Bioconductor have been developing. And there's scholarships for graduate students to attend the 2020 Cold Springs Harbor Lab Biological Sciences Conference that are due August 1st that you can find references to on the internet. With that, I'll stop sharing, and I think that we've run out of time actually, and we need to end the session. So I'll do that, I guess, and look forward to meeting you all at different opportunities during the conference. Thanks for joining.
Thanks, Martin.